Today we are going to discuss a new topic named Pseudomonas. And earlier we have discussed different bacilli and Bacteriaceae in which we studied Shigella, Salmonella, E. coli, etc. And they all are gram-negative bacilli. And again, this Pseudomonas and describes a large group of aerobic, non-fermentative, non-sporing and gram-negative bacilli, and they are motile by a polar flagella. Okay, and the genus this Pseudomonas belongs to the family of Pseudomonaceae, and which contains over 200 species, and out of which today we are going to discuss the Pseudomonas aeruginosa. And this is medically important and most commonly associated with human diseases. Okay, so we will discuss about the morphology of this Pseudomonas aeruginosa. First of all, we see they are cylinder, gram-negative bacillus, and the measurement, if we see the measurement, they are 1.5 to 3 micrometer in length, and the breadth is 0.5 micrometer. Okay, and they are actively motile and usually with a singly polar flagella, and the occasional strains have 2 or 3 flagella, and the fimbriae may be present and are usually polar and non-hemogglutinin. And again, this Pseudomonas aeruginosa is non-sporing, non-capsulated, but many strains have mucoid slime layers. Okay, and this is all about the morphology of this Pseudomonas aeruginosa. Now we will discuss about the cultural characteristics. Okay. Again, it is a strict aerobic and grows well on ordinary media like nutrient broth, nutrient agar, and the growth occurs at a wide range of temperature, and the temperature range is 6 to 42 degrees centigrade, and the optimum temperature will be the 37 degrees centigrade in different bacilli, and the optimum pH will be the 7.4. pH will be the 7.4 to 7.6, and this is the general aspect of this cultural characteristics. And now we see the nutrient agar and Maconchi agar. Next is blood agar, broth, that is petrol water, and the selective media, we will study the selective media, and that is citramide, citramide media or agar. Media or that will be the agar. And when see this nutrient agar, and after aerobic incubation on this nutrient agar at 37 degrees centigrade and for 24 hours, the colonies are large, 2 to 3 millimeter in diameter, and the colonies will be seen very smooth, translucent, low convex, irregularly round, and emit a sweetish aromatic odor. And in other words, we can also say fruity odor. इसमें से फल जैसी एक गंध आती है. And there is a greenish blue pigment which diffuses in the medium. ये ग्रीन सा एक पिगमेंट होगा, ग्रीनिश ब्लू, और जो पूरे मीडियम में डिफ्यूज फैल जाएगा. एंड व्हेन वी डू द इनोकुलेशन ऑन मैकोंकीज agar, and in this we see the non-lactose fermenting colonies, that will be seen in pale or colorless. And when we inoculate on blood agar, and the colonies may be surrounded by a zone of hemolysis, and other characters are similar to this nutrient agar. So colonies may be large, smooth, translucent, and low convex, is regularly long, and they emit a sweetish aromatic odor. And when we inoculate this specimen in petrol butter, it forms a dense turbidity with surface pellicles. Okay, and next, when we use the citramide agar as selective, it is a selective medium. In some cases, we use the citramide agar. Citramide agar medium भी कह सकते हैं और agar भी कह सकते हैं. ठीक है, these are the agars or medias on which we culture this Pseudomonas aeruginosa. Now we will talk about the pigments, which we see that there are different type of pigments, product, pigment, production. As we have seen that there are different type of pigments are produced by this Pseudomonas aeruginosa, and they diffuse in the surrounding media. And there are now we will discuss there 4 pigments. There are 4 pigments we are going to discuss. That is pyocyanin, and second is pyoverdin, and third one is pyoruberin, and fourth one is pyomelanin. These are the 4 type of pigments which are produced by this Pseudomonas aeruginosa while we are culture. And when this pyocyanin is bluish green pigment, and this is soluble in chloroform and water. And this pigment is not produced by other species of this genus, and hence it is a diagnostic of this Pseudomonas aeruginosa. And this pyoverdin, second number, this pyoverdin, है ना, ये अपन लिख देते हैं bluish green. And this pyoverdin is yellow green. Yellow green pigment, and it may be produced by many other species also. And this next pyoruberin, and it is bright red, and it is also soluble in water, and it is phenazine pigment that is insoluble in chloroform. And when we talk this biomelanin, and it is brown to black pigment, colored pigment, and its production is uncommon, and it is chemically unrelated to the animal melanin. So again, this Pseudomonas aeruginosa produces different type of pigments which diffuses in the medium. The name are pyocyanin, that the bluish green color, pyoruberin, yellow green color, pyoruberin, bright red color, and pyomelanin is brown to black color. Okay, and now we will talk about the cultural characteristics here, and next is biochemical reactions. Biochemical reactions, and this differs from the other members of activated by deriving energy from carbohydrates by an oxidative breakdown rather than a fermentative metabolism. Okay, हम आपका fermentative metabolism के देख रहे थे, होता है यहाँ पे हमारा oxidative breakdown होता है, and it utilizes only glucose oxidatively with acid production. There will be no gas, है ना? And lactose and maltose are not utilized, and indole, methyl red, and the VP, that is Vorscos porpor, and these hydrogen sulfide are negative. These tests will be found in case of this Pseudomonas aeruginosa is negative. And while catalase and oxidase are positive. Okay, na? And this reduces the nitrates to nitrites, and further to gaseous nitrogen. Okay, so this is all about this biochemical reactions which occur in Pseudomonas aeruginosa. And now we will discuss about the resistance and viability of this Pseudomonas, and this bacillus is killed at 55 degrees centigrade in 1 hour, and resistant to common antiseptics and disinfectants such as quaternary ammonium compounds, chloroxylidol, and hexachlorophene. These are the common disinfectants and antiseptics which are used in our hospitals and daily used in laboratories. And this bacillus is sensitive to 2% aqueous alkaline solution of glutaraldehyde, and that is also known as Silex. And this Pseudomonas aeruginosa possesses a considerable degree of natural resistance to antibiotics. मतलब ये कुछ एंटीबायोटिक्स का इसके ऊपर कोई प्रभाव नहीं पड़ता है, वो रेजिस्टेंट होता है। सो दिस इज ऑल अबाउट दिस Pseudomonas aeruginosa, and the next part of this, that is pathogenesis and laboratory tests, we will study in next lecture. So thank you.